What was the first organism like? This is an age-old question and considered one of the many holy grails of science. There are many competing hypotheses out there, but here I'm going to give my take on what the first organism was like. The time is roughly 4 billion years ago at the Archean-Hadean boundary. The Earth is largely a water world, but there are a few scattered islands. These islands are composed of mafic and ultramafic rocks, the latter of which includes olivine and pyroxene. Here, rainwater collects into small pools which then chemically reacts with the ultramafic rocks in a process known as serpentinization, which releases hydrogen gas. In the periphery of these pools is montmorillonite clay. Within a pool is a lipid vesicle composed of simple carboxylic acids. Contained within this vesicle is a folded RNA polymer. This polymer is known as a ribozyme, which carries out functions similar to enzymes. This one in particular is a dual-function kinase-ligase ribozyme. The lipid vesicle, or protocell, also has short RNA oligomers floating around within it. The ribozyme quote-unquote replicates itself by combining these short RNA oligomers into a completed polymer. So, each individual RNA oligomer is a piece of the polymer. How does it do this? First, it binds to one of the free-floating oligomers via complementary base pairing, and then reacts the phosphate group on the nucleotide on the 5' end of the oligomer with either a pyrophosphate or metaphosphate ion, making it a diphosphate nucleotide. Where do the pyrophosphate or metaphosphate come from? Meteorites fall regularly, and the metallic ones have schreibersite inclusions within them, and when immersed in water, this iron-nickel phosphide will produce reactive phosphorus species like pyrophosphate and metaphosphate. So, now the 5' nucleotide is activated, and it can be joined to another sequence. This is then repeated until a new ribozyme is assembled. But how are these short RNA oligomers produced? Being a small terrestrial body of water, the pond will regularly dry out and refill with rainwater. When the nucleotides dry on montmorillonite clay, they will polymerize, and when contained in a lipid vesicle, they don't even have to be activated to do so, and the existing oligomers can act as a template. The template and nascent strand will be bound to one another via hydrogen bonding. But the serpentinization reaction induces an alkaline environment, and due to the carbon dioxide in the atmosphere, rainwater will have a pH of around 6. So rainfall induces pH variations in the pool, and this acidic alkaline cycling allows the RNA strands to separate. But where do the nucleotides come from? Below the pool, hydrogen produced by the serpentinization reactions reacts with carbon dioxide from the atmosphere, which results in the production of simple organic molecules. Some of these are carried up into the pond by convection currents and cross the membrane into the protocell. As mentioned before, the ribozyme is a dual-function ligase/kinase. Often the activity of an enzyme or ribozyme can be altered by variations in temperature and pH. These variations can tweak their catalytic mechanisms, allowing them to facilitate similar reactions. And since ligation reactions are phosphotransfers, it wouldn't be a huge leap to catalyze kinase reactions where a phosphate group is bound to an organic molecule. Once some of these simple organic molecules, such as glyceraldehyde, are phosphorylated, they can be fed into abiotic analogs of various metabolic pathways such as the pre-pentose phosphate pathway. Instead of being catalyzed by enzymes or even ribozymes, this reaction network is catalyzed by magnesium ions and iron 2. This produces ribose, and when borate is present, it will be reacted with an orthophosphate and urea to form a prenucleotide. Then, other nitrogenous organic molecules are added to the urea to form a nitrogenous base, be it adenine, guanine, cytosine, or uracil, completing the nucleotide. However, these magnesium ions are chaotropic, meaning that they will regularly break down cell membranes. Thankfully, amino acids are floating around in this mixture, which binds the ions acting as chelating agents. This allows the protocells to have their cake and eat it too, i.e., use the catalytic properties without having the divalent metal ions break down the membrane. Amino acids can also act as scaffolding agents, stabilizing the RNA polymers. Where do these amino acids come from? In our cells, the Krebs cycle is mostly used to oxidize organic carbon to produce energy. However, intermediates, known as alpha-keto acids, act as the precursors of amino acids. These then have an amino group bound to them, thus becoming an amino acid. Abiotic analogs of the TCA cycle have been observed catalyzed by iron-sulfur minerals in hydrothermal systems. In our cells, the TCA cycle is run oxidatively or clockwise by feeding oxidized electron carriers into it, which shuttle the electrons to enzymes that transfer them ultimately to an oxygen. However, some bacteria run it reductively or counterclockwise by feeding reduced electron carriers into it. Whether the abiotic analog would run oxidatively or reductively would depend on how oxidizing or reducing the surrounding environment was. The oxidants or reductants can be organic or inorganic, i.e., they can be highly oxidized or reduced organic molecules, or they can be hydrogen or perchlorates. Strecker reactions bind ammonia in the solution to the alpha keto acids to produce amino acids. These reactions are catalyzed by either copper, nickel, cobalt, or vanadium ions. In order for this abiotic TCA cycle to produce alpha keto acids, it has to be fed simple organic molecules, most notably glyoxylate and acetate. These small organic molecules are again produced by subsurface Fischer-Tropsch type reactions, and, like the abiotic TCA cycle, these are catalyzed by iron sulfide minerals, most notably greigite and mckenowite. Metasedimentary rocks from 3.8 billion-year-old mud volcanoes are replete with ammonium ions, implying that ammonia was present in the solution. However, where it came from is a bit of a mystery. In a manner similar to Fischer-Tropsch type synthesis reactions, the Haber-Bosch process produces ammonia from hydrogen and diatomic nitrogen. These reactions are again catalyzed by metals, but since the triple bond binding the atoms of a diatomic nitrogen molecule are much stronger than the double bonds that bind the carbon dioxide molecule, it requires temperatures and pressures higher than those found in naturally occurring hydrothermal systems. The interior of the young Earth had a much higher concentration of radioactive elements than the modern Earth, and it's been hypothesized that the high-energy particles released by the decay of these elements could break the triple bond of a nitrogen molecule so it could be reduced to ammonia. But in my opinion, the answer lies not below but above. The young sun was more temperamental than it is now, and would produce flares with solar wind intensities hundreds of times greater than what we see today. The energy in these particles could then break the bonds in atmospheric nitrogen molecules, producing nitrogen radicals, which can then be reduced to ammonia in hydrothermal settings. While most of the organic molecules produced by Fischer-Tropsch synthesis reactions will be simple 1, 2, or 3 carbon molecules, some will be elongated to form fatty acids. Furthermore, montmorillonite clay can catalyze the production of fatty acids from smaller organic acids and alkanes, such as formate and ethane. These can then be included in the lipid membrane of the protocell, facilitating its growth. Most lipid vesicles will be in equilibrium with their environment, meaning that they are gaining and losing fatty acid molecules to and from the solution at the same rate. However, when RNA nucleotides are polymerized, they increase the osmotic pressure within the vesicle. This causes it to collect more fatty acid molecules than it loses and grow. Eventually, it will become large enough that shearing forces caused by wave action and convection currents break apart the vesicle into smaller daughter vesicles, many of which inherited the RNA oligomers and polymers from the parent vesicle. But apart from taking organic molecules in the environment to be synthesized into biomolecules, is there a way that our protocell could produce some of its own? Well, you'll have a bunch of mineral nanocrystals floating around in solution, which will normally precipitate out joining larger crystals. However, phosphorylated organic molecules can react with the sulfide groups on metal sulfide minerals, forming thiol groups. If the organic molecule is hydrophobic, the nanocrystal will become embedded in the membrane. So, if it's an iron-sulfur nanocrystal, it can then act as a catalytic surface for the abiotic TCA cycle, producing amino acids for the protocell. But can we go a step further and get the first organisms to be autotrophic at this early stage? Well, possibly. As mentioned before, Fischer-Tropsch type synthesis reactions produce organic molecules in natural settings, but occur at temperatures and pressures too high for anything to survive. So, to see if we can get it to work under biologically relevant conditions, let's crunch the numbers behind the reaction kinetics. These reactions are catalyzed by metal sulfides in hydrothermal systems, most notably iron sulfides, but to a lesser extent nickel sulfides. At pressures around 1 atmosphere, and at temperatures less than 100 degrees Celsius, ferrous iron can't oxidize hydrogen, and zero-valent or metallic nickel can't reduce inorganic carbon. Nickel too can oxidize hydrogen, and a metallic iron can reduce carbon, but there's a catch. Nickel will only do so at high pHs, while iron will only do so at neutral and acidic pHs. Is there a way around this conundrum? Well, yes. It's been experimentally demonstrated that if you have an alkaline hydrogen-rich solution separated from an acidic inorganic carbon-rich solution by a semiconducting iron-sulfur-nickel barrier, bicarbonate reduction will occur. So basically, a nickel oxidizes hydrogen, and since it's in a cluster, the electrons get sloshed around between the iron and nickel atoms. So some of the iron atoms on the acidic side will be reduced to zero-valent iron, which can then reduce bicarbonate to formate. More hydrogens can then be oxidized to further reduce the formate to a formyl, methanol, and finally a methyl group. If there are also cobalt impurities, the methyl groups are more likely to be added to another carbon being reduced, forming a carboxylic acid to be incorporated into the membrane. There is some evidence for this. The Wood-Young-Dahl pathway used by acetogens is mechanistically similar to the abiotic Fischer-Tropsch style reactions. Now you might think, hold the phone, this would require a cross-membrane pH gradient, and these need complex molecular machinery to maintain. Well, this environment will be subjugated to changes in pH, and because a lipid membrane is less permeable to ions such as protons and hydroxide ions than it is to water molecules, this means the membrane will have a buffering effect, and should the external pH quickly go from neutral to basic, the internal pH would, for a time, remain neutral. So, we do have a temporary cross-membrane pH gradient, which can then allow protocells to produce their own organic molecules from inorganic carbon, i.e. be autotrophic, even at this early stage. So, to recap the cell cycle of this primitive organism, RNA oligomers are assembled into a ribozyme by a pre-existing ribozyme. The ribozyme then phosphorylates small organic molecules after an alternation in the temperature or pH, which ultimately leads to the synthesis of nucleotides. These simple organic molecules can be either obtained from the surrounding environment or possibly produced within the cell. The water in the pool dries out, leading to the production of more RNA oligomers. When the pool refills with water, these oligomers increase the osmotic pressure on the membrane, causing it to take more lipids from the environment, although the cell may again be able to produce some of its own, until it gets broken apart by mechanical forces and the cycle repeats. Of course, since these are driven by external processes, events won't always happen in this order, so the life cycle of the first cell will be nowhere near as orderly as that of modern cells. It's often said that cells have three key properties: metabolism, compartmentalization, and reproduction. And this protocell has metabolism mostly driven by inorganic metal ion and mineral catalysts, compartmentalization provided by the carboxylic acid membrane, and replication facilitated by the ribozyme, which acts as the primordial genome. This brings me to a concept called the minimum Darwinian threshold. The origin of life is often called the transition from chemistry to biology, but the problem I have with this description is that biology is ultimately governed by chemical processes. A better description would be the transition from geochemistry to biochemistry. We're all familiar with the role of a catalyst, or a chemical that lowers the activation energy of a reaction. What makes life distinct from most other chemical reactions is that it is autocatalytic. This is where the product of a reaction also acts as its catalyst. We catalyze a chemical reaction, i.e. the oxidation of organic carbon. But while the major products of this reaction are carbon dioxide and water, the biomolecules which constitute our tissues are as well. So, we are both the catalyst and the product of this reaction. When you have a mix of reactants and plot the concentration of products over time, the rate of the reaction will start out high, but slow down as the reactants run out. Adding a catalyst will shift this curve over to the left, increasing the rate of the reaction at all concentrations. However, if you have an autocatalytic reaction, the rate will start off slow and increase over time as the increased concentration of the product catalyzes the reaction, causing it to run faster. But with a given amount of reactants, eventually they will start to run out, so the concentration of the product will hit an inflection point. After this, the rate of the reaction will slow down as the quantity of the reactants approaches zero. To clarify, a reaction being autocatalytic doesn't make it alive by default. I mean, if you initiate an autocatalytic reaction network in a pool of reactants, the reaction will spread out. But if you also ignite a fire on a field of combustible materials, it will also expand outwards. But this doesn't mean that the fire is alive. If we define the origin of life as the minimum Darwinian threshold, you don't just need a self-replicating reaction. You need one whose catalyst is mutable to change to become either better or worse at catalysis. The autocatalysts, which become modified to act as better catalysts, will consume reactants faster, produce the product, i.e. itself, at a faster rate, and thus outcompete other catalysts. This is why something has to have inheritable features to be considered alive and be subjected to Darwinian evolution, and hence meet the minimum Darwinian threshold. Proponents of the metabolism-first view of the origin of life, which postulates that the first cells didn't have genetic material, have argued that the first organisms passed information from one generation to the next via their chemical composition, also known as their compositional genome or composome. However, simulations have shown that the heritability of such systems is too low to allow for Darwinian evolution. In The Origin of Life, the Geochemical Roots of Biology series, I argued that the first cell had a ligase ribozyme, but now I think this ribozyme would have to have some kind of kinase activity as well to meet the minimum Darwinian threshold. And to clarify, this is just my opinion. I haven't run any simulations or experiments to back this claim, nor can I cite any. By carrying out kinase activity, the ribozyme can initiate the production of more of its reactants, i.e. nucleotides. This will make protocells which contain this ribozyme much less limited than other autocatalytic reaction networks. The reason this is so critical is that since RNA is much less stable than DNA, it has to be replicated at a greater rate to be inheritable, and should a self-replicating sequence run out of nucleotides, the polymer will become hydrolyzed before it can self-replicate, and the genetic information will be lost. This would link the genetics of the first organism to its metabolism, and in so doing, lay the foundation for the central dogma of molecular biology. Finally, the osmotic pressure induced by the polymerization reactions would tie the replication of genetic material to the growth and division of the cells. As discussed earlier, oftentimes enzymes and ribozymes can catalyze reactions that have similar mechanisms. In fact, this is what often allows enzymes to evolve quote-unquote novel functions. However, this flexibility comes at a cost. These dual-purpose biocatalysts are not as efficient at catalyzing each individual reaction as more specialized ones. As such, this ancestral ribozyme could have evolved into two descendant varieties, one being a kinase, while the other was specialized for ligation. This would give any protocells with these ribozymes the ability to both produce nucleotides and assemble RNA polymers at greater rates, allowing those with these new varieties to outcompete those who only had the original ribozyme. A third variety of ribozyme, a polymerase, which can assemble sequences in solution from free-floating nucleotides using an existing oligomer as a template, can then evolve from the specialized ligase. Now, the early RNA oligomers can be produced in situ versus by drying on montmorillonite clay. Since the production of RNA polymers is tied to growth and division, this would give organisms with this polymerase an advantage over those that didn't, again allowing them to outcompete the precursors. So now we can see Darwinian evolution at work, facilitating the development of more intricate systems. Where it went from there is a topic for another video, but now that we've kickstarted evolution by natural selection, the sky is the limit. Well, that's my best guess as to what the first organism was like. Abiogenesis is of course an ongoing field of study, so new discoveries can completely change our understanding of the origin of life in the near future. But, just to give you an idea of the scope of this question, Let's say someone produced a viable organism from non-living material in a lab. While such an achievement would surely be worthy of a Nobel Prize, it wouldn't end the controversy. Remember, just because we can make a de novo organism under one of the myriad of conditions present on the prebiotic Earth, that still doesn't prove that's how life began. Hey everyone, I've noticed this channel has been growing a lot lately, so I figured I'd give a little outro to discuss future plans. As a kind of sequel to this, I'm planning a video about the early evolution of biocatalysts and genetics, followed by one about the origin of modern bioenergetics and carbon metabolism, then another one about the origin and evolution of respiration and photosynthesis, and finally I'll wrap up the early life series with a video about the ecology of the Archean Earth. I've already touched on these topics a bit in the Stem of Life and Before the Great Oxygenation Event series. Also, if you want to know more about the geochemical processes taking place on the early Earth, you can see the Origin of Life series. Another video I'm currently working on is life on red dwarf planets, but some other topics I hope to cover include recognizing alien intelligence, microbial ecophysiology, the alternative biochemistry iceberg, gravity and life, and the scientific accuracy of Avatar. Between those, I'll probably also be making a few random shorts as well. If any of these topics are of particular interest to you, let me know in the comments. Lastly, if you follow me on Twitter or X, I don't just post my videos there. If I post a comment on a video, I'll post it there as well. And if I find an interesting publication, I'll post it and maybe add some commentary. So that's all for now. If you like what you see, like and subscribe. It helps the algorithm a lot. And thank you again for all your support.